The ability of living organisms to respond and to communicate with their environments has grown in parallel with the evolution of their nervous systems. The earlier systems were peripheral, like this surface nerve net in the foot of Hydra. In the flatworm, with its nerve cord and brain-like head ganglion, the move towards a central system is clear. A rudimentary brain, spinal cord and spinal column appears in primitive chordates like Amphioxus. And in these unusual animals, neither invertebrates nor vertebrates, the tubular beginnings of the human nervous system can already be recognized. At 2 weeks, the embryo consists of 2 layers, endoderm and ectoderm. By the middle of the 3rd week, a 3rd layer, mesoderm, appears between them. The brain and spinal cord develop from ectoderm. A central area thickens to form a plate of neuroectoderm called the neural plate. Within it appears a shallow groove, the neural groove. The groove is flanked by folds called neural folds. Neuroectodermal cells from the lateral edges of the neural plate form an intermediate layer called the neural crest. The neural crest gives rise to a variety of nervous tissue, such as ganglia, and non-nervous tissue such as the inner 2 layers of meninges, the arachnoid and pia mater, which together comprise the leptomeninges. With further growth, the neural folds fuse in the midline to form the neural tube. From its cells develop not only neurons, the excitable cells of the nervous system, but also some types of neuroglia, the supporting cells of the central nervous system. Astrocytes and oligodendrocytes, for example, develop from ectoderm. Microglial cells develop from mesoderm. Fusion of the neural tube begins in the cervical region and continues cranially and caudally. The caudal third of the neural tube develops into the spinal cord. At first, the wall of the neural tube comprises a single layer of neuroepithelial cells. Cells in the lateral walls then divide rapidly to form 3 zones: ventricular, intermediate, and marginal. The ventricular zone gives rise to ependymal cells which line the central canal of the spinal cord. The intermediate zone, which ultimately forms gray matter, initially comprises primitive nerve cells called neuroblasts. The outer marginal zone consists of their processes, the axons and dendrites, which after myelination form white matter. As the lateral walls proliferate, they become partially separated by a shallow groove called the sulcus limitans into basal and alar laminae. Neuroblasts in the basal lamina form the motor cells of the anterior and lateral gray horns, while those of the alar lamina form the sensory cells of the posterior horn. The cranial end of the neural tube expands to form the brain. It develops from 3 primary vesicles: the forebrain, the midbrain, and the hindbrain. Initially, the primitive brain comprises the same 3 zones as the spinal cord: ventricular, intermediate, and marginal. As in the spinal cord, cells of the intermediate zone form an inner layer of gray matter. In the brain, some of the cells migrate through the marginal zone to form a second outer layer of gray matter, the future cerebral cortex. As it grows, the brain folds. The midbrain flexure, between the midbrain and the forebrain, appears first. The temporary cervical flexure, between the hindbrain and the spinal cord, next, and last appears the pontine flexure, at the level of the future pons.